Hello students, I am Prithwi. Today we are going to discuss about gene promoters. In today's discussion, we are going to learn about prokaryotic promoters, various types of prokaryotic promoters used in expression vectors, and how to select a prokaryotic promoter for gene cloning. This is a pictorial representation of a gene. What is a gene? A gene is a sequence of DNA which is transcribed into RNA and contains mainly three important parts. That is the promoter, the coding sequence, and the terminator. Now, what is the promoter? It is the non-coding sequence responsible for the initiation of transcription, right? That is, a promoter is the region of the DNA where transcription of a gene is initiated. And promoters are a vital component of expression vector because they control the binding of RNA polymerase to DNA. And we know that RNA polymerase transcribes DNA into mRNA and which is ultimately translated into a functional protein. Now, before we learn more about promoters, we should know about the structure of the E. coli, that is the prokaryotic RNA polymerase. This is the typical structure of E. coli's RNA polymerase. Here you can see that E. coli has only one RNA polymerase and it is made up of 6 subunits, that is 6 different kind of polypeptides. 2 alpha, that is alpha 2, beta, beta prime, omega and sigma. All these 6 subunits together make up the holoenzyme. However, this RNA polymerase without the sigma is called the core enzyme. And we know that the RNA synthesis, that is the transcription, consists of 3 different phases, that is initiation, elongation and termination. We can see a ribosome binding site. Actually, a ribosome binding site is a segment of the 5' upstream part of an mRNA molecule that binds to the ribosome to position the message correctly for the initiation of translation. The RBS controls the accuracy and efficiency with which the translation of mRNA begins. It is also known as the Sine-Dalgarno sequence, which is composed of the polypurine sequence located just 5' of the AUG initiation codon. The sigma subunit of prokaryotic RNA polymerase recognizes consensus sequences found in the promoter region upstream of the transcription start site. The sigma subunit dissociates from the polymerase after transcription has been initiated. In prokaryotes, the promoter consists of 2 short consensus sequences, one at -10 and another at -35 position upstream of the transcription start site. The consensus sequence at -10 is known as -10 element and usually consists of the 6 nucleotides TA, TA, AT. This element is essential to start transcription in prokaryotes. There is another consensus sequence at -35, which is also known as -35 element, usually consists of 6 nucleotides, that is TT, GA, CA. Its presence allows a very high transcription rate. So both these two elements, that is -35 and -10, are essential for prokaryotic transcription. The promoter is the most important component of an expression vector. This is because the promoter controls the very first stage of gene expression, that is, the attachment of an RNA polymerase enzyme to the DNA, and determines the rate at which mRNA is synthesized. Therefore, the amount of recombinant protein obtained depends to a great extent on the nature of the promoter carried out by the expression vector. Here we can see the pictorial representation of our expression vector, and a specific gene is cloned within the expression vector, and the vector is transformed into E. coli for gene expressions. Most of the E. coli promoters do not differ much from the known consensus sequence we have discussed in the previous slide, and the small variations may have a major role on the efficiency with which the promoter can direct transcription. In this slide, we will discuss about two kinds of promoter, that is a strong promoter and a weak promoter. Strong promoters are those that can sustain a high rate of transcription. Strong promoters usually control genes whose translation products are required in large amounts by the cell, whereas in contrast, weak promoters are relatively inefficient, direct transcription of genes whose products are needed in only small amounts. Clearly, an expression vector should carry a strong promoter so that the cloned gene is transcribed at the highest possible rate. There is a pictorial representation of the two types of promoter, that is a strong promoter and a weak promoter. And in the picture we can clearly see the strong promoter produce a large amount of transcript which are translated into numerous protein molecules, whereas in comparison the weak promoter produce relatively few transcripts, resulting low number of protein molecules. A second factor to be considered when constructing of an expression vector is whether it will be possible to regulate the promoter in any way. Two major types of gene regulation are recognized in E. coli. One is induction and another one is repression. An inducible gene is one whose transcription is switched on by addition of a chemical to the growth medium. Often, this chemical is one of the substrates for the enzyme coded by the inducible gene. In contrast, a repressible gene is switched off by addition of the regulatory chemical. In this picture, we can see an inducible gene, right, which is induced only by the addition of a regulatory chemical, whereas we can also see a repressible gene. That is, the expression of the repressible gene switches off the gene expression. In this slide, we will discuss about various kinds of promoter used in expression vectors. Let's start with the lag promoter. The lag promoter is the sequence that controls transcription of the lag Z gene coding for beta-galactosidase. The lag promoter is induced by IPTG, so addition of this chemical into the growth medium switches on transcription of a gene inserted downstream of the lag promoter carried by an expression vector. The next is the tryptophan promoter. The E. coli TRP promoter has been extensively used for the high-level production of proteins on a small and large scale. This regulated promoter is readily available, relatively easy to turn on, and can be used in essentially an E. coli host background. The TRP promoter is normally upstream of the cluster of genes coding for several of the enzymes involved in biosynthesis of the amino acid tryptophan. The next is an interesting promoter, the tag promoter. The tag promoter is a hybrid promoter. The tag promoter is a hybrid between the TRP and the lag promoters. It is stronger than either but still induced by IPTG. The last is the T7 promoter. The T7 promoter is specific for the RNA polymerase coded by T7 bacteriophage. This RNA polymerase is much more active than the E. coli RNA polymerase, which means that a gene inserted downstream of the T7 promoter will be expressed at a high level. The gene for the T7 RNA polymerase is not normally present in the E. coli genome. So what we need to do, so a special strain of E. coli is needed, one which is lysogenic for T7 phage. In this slide, we'll learn how to select a prokaryotic promoter. What I have done in this slide, I have selected a gene sequence from Sinorhizobium meliloti 1021 database, and we can see that the gene starts with the start codon and ends with the stop codon. So where is the promoter? Generally, for selecting prokaryotic promoter, we selected 200 base pair upstream of the start codon, and this is the start codon, and the region which is not highlighted in yellow is the promoter. So this is a putative promoter. Now what we need to do, we need to use any software or online tools available to see whether this is a true promoter, and even it's a promoter, how good is that promoter. We are going to use bprom for the prediction of bacterial promoter. bprom is a bacterial sigma 70 promoter recognition program with about 80% accuracy and specificity. It is best used in regions immediately upstream from the open reading frame. bprom is a free web tool. We can access it online. Even we can see there is a space where we can paste the nucleotide sequence. What we'll do, we'll copy the 200 base pair region from the upstream of the gene. Please refer to the previous slide in FASTA format, and we'll wait for the result. In this slide, we can see that we have already pasted the sequence in FASTA format, right? And after that, what we'll do, we'll just click on the process button and we'll have the result. Here is the result, and we can see that the number of predicted promoter is 1, and our promoter, the promoter we have selected, has a -10 consensus sequence and a -35 consensus sequence. So that's very interesting, and we can do some other promoter analysis using the software bprom. You can select various genes from the available database of various prokaryotic organisms, for example, E. coli genome database or Sinorhizobium meliloti 1021 database, and select around 200 or 300 base pair or more than that the nucleotide sequences from the upstream of the gene, and you can try to score them. You can try to find out how good is the promoter, which one is the best promoter, and which one is not that good. And we can also see there is a score. So the promoter we have selected, it scores around 7. It may be possible that we can get some promoters having a more better score than 7, or maybe a bad score than 7. So for that, we need to select various genes, their upstream sequences, and just use the bprom tools and try to find out whether they are two promoters, and if they are promoters, how effective they are. Thank you for watching the video, and I hope we have learned a little bit about prokaryotic promoter.